Alright folks, well this is my first attempt at this, so forgive me if this gets rambly. You can go ahead and pause anytime you need to. And today what we're going to be doing is we're going to be covering the process of respiration. And that would be getting energy out of glucose molecules, essentially the reverse process of photosynthesis. One of the early diagrams in the chapter of the book discusses how light energy comes in, energizes these chloroplasts, and they result in organic molecules and oxygen being produced. That in turn, those organic molecules, that glucose is used in mitochondria, and through that process, which we'll talk about, we create carbon dioxide and water, which then can be fed back to the chloroplasts. Overall, when we respire, we make ATP, which is used to make energy for the cell or power individual things going on inside the cell. And as a result, you create entropy back to the universe. One of the earlier discussions within the text is also about redox reactions, reduction and oxidation. Easy thing to remember is Leo the lion says grr, and Leo would be LEO, loss of electrons is oxidation. So in this example here, sodium is losing an electron, becoming positive. Chlorine is gaining an electron and becoming negative. So GER, G-E-R, gain of electron reduction. One of the main players in this entire process is NADH, and in that context we have an NAD plus molecule over here, and this would be NADH over here. We added on an H. The gain of electrons, remember, is reduction, and the loss of electrons going back this way would be oxidation. So this is the oxidized state, this is the reduced state here. Keep that in mind. NADH and NAD plus are kind of interchangeable. Depends on how they're carrying around electrons. One more thing before we get going here. The book also illustrates, talks about how certain chemical reactions will happen just kind of spontaneously or are favorable for energy, and they end up in a explosive release of energy. The whole idea behind cellular respiration is that they want to break down each individual step along the pathway to harness little bits of energy along the way, which then can be used with enzymes to convert all that little incremental energy into molecules of ATP. So that's the overall idea behind cellular respiration, is having a controlled reaction here versus an uncontrolled reaction over here. All right, so there's some main pathways here that are covered in the overall idea. And the main pathways are going to be, I'll back up here, the main pathways are going to be glycolysis is your first one, and that results in some ATP and electrons carried via the NADH molecule. The second pathway is citric acid cycle, and again we're creating electron carriers and some ATP. And then the final pathway here is what's referred to as the electron transport chain and chemiosmosis, which would be creating ATP. All right, here's some questions for you. You can go ahead and pause your presentation and see if we can figure out some of the answers to these questions. I'll start again in about 3 seconds. All right, here's a reminder of the overall concept of respiration. Again, we're trying to break apart glucose into usable forms of energy of ATP in stepwise fashion. The first part of this would be glycolysis. And the book has a very detailed diagram here of these things going on. Now, what needs to be recognized here is that this is called the energy investment phase. Glucose itself is a very stable molecule. If you recall, we talked about destabilizing molecules in class by adding ATPs to it, which would add a phosphate to this whole mess. So we'd add a phosphate once, and we'd add a phosphate twice through 2 ATP molecules. And what that does is it really destabilizes this molecule, and this molecule essentially just cuts itself right in half. Overall, this is kind of counterintuitive because you need to consume energy in order to get energy back out of this. So again, it's kind of an investment. It's going to pay off down the road. Cells need to do this, and in order to create these 2 different forms, which are isomers of each other, and eventually this one goes on in the whole chain. So if we create this one, it automatically converts into this and then continues on into the whole chain. Again, the idea is that you put in 2 ATPs and that we are rearranging things. Then we have the payoff phase, which is this series of steps here. Now what we have to do is, I'm going to go back one slide. You have to realize that we have created 2 molecules of this stuff, essentially. 2 molecules of this stuff, because we cut this thing in half, we have 2 3-carbon molecules. If this is formed, this converts into this, and we're left with a second one of these. So that's kind of the idea behind it. Overall then, what you need to do is, to get energy out of this whole glycolysis process, we use 2 NAD plus molecules, which pick up electrons and hydrogens from that molecule before. We use 2 of them because there were 2 of them at the end of the process, and we end up with 2 NADH molecules being created, pulling a hydrogen off the whole mess, and you end up with 2 of these. That's why they have like a little overlaying 2 here. You see that there's like 2 different panes there behind each other. Again, if we consider keep moving down the road now this thing is very unstable and able to hand back off the phosphate that was attached in the first place. We get 1 of them back, and because there's 2 of them, we get a second one back. Then we end up with this thing, which rearranges and rearranges, and eventually we can pull off 2 more phosphates off of that molecule. So overall, we had to invest 2 ATPs, and we got 4 ATPs back out of it. The final product here is called pyruvate. Not sure how well you can see that, but it's pyruvate. You can check your book for that one. Pyruvate, it's a 3-carbon molecule, and keep in mind we have 2 of them. So 1, 2. All right, here's some checkpoint questions for you. I'll go ahead and let you pause this for 1 second. Go ahead and use your book if you need to. I'll pause for about 3 seconds, and you guys can catch up when you need to. All right, going back to the overview again here. Our first step was glycolysis, where we took glucose and chopped it in half and made 2 pyruvate molecules. In the process, we had to invest 2 ATPs. We got 4 ATPs back out of it, and we created some NADH molecules, which will be used later on. Then we're going to move into the citric acid cycle. The whole idea here is, before we move into the citric acid cycle, realize that this happens outside in the cytosol of the cell, the liquid medium of the cell. And we create the pyruvate out here. Once that pyruvate is created, it needs to go inside the mitochondria, and that would be the mitochondria, or the powerhouse of the cell, as you probably learned. Once that pyruvate transfers across that membrane, there's a little process that happens, and what goes on here is that we are able to take off of pyruvate, we're able to take a CO2 molecule. Now keep in mind there are 2 pyruvate molecules for every glucose that started up, so we really remove 2 CO2s from the process. Now we have 2 2-carbon molecules that are going across this membrane. Once they're in, NAD plus becomes a reducing agent and becomes NADH, and coenzyme A joins in, and it attaches to this molecule, making it a little bit more stable. Without the coenzyme A, this molecule is just an absolute mess and does not stabilize very well. So coenzyme A attaches to it. Now it's ready to do its job. Its whole job is to go into the citric acid cycle and go through a whole series of steps. This is going to look a lot like the Calvin cycle, so keep that in mind. Now here's that whole Krebs cycle, or citric acid cycle, or you may hear it a couple of different ways, but ultimately what's going to happen here is a whole series of steps. Again, I told you it looks like the Calvin cycle a lot. Here's your acetyl coenzyme A coming in. Remember, we have 2 molecules of this. This goes through at once, so we'll clarify that in a second. So for every 1 molecule, what happens is that joins up with this molecule here, oxaloacetate. That oxaloacetate has 4 carbons in it. Those 4 carbons then are joined with the 2 carbons, and we end up with a 6-carbon molecule called citrate. Citrate goes through a series of steps. Along each step is an enzyme that does these jobs. And what they do is that they keep reducing NAD plus into NADH. Remember, a gain of electrons is reduction. They also strip off carbons, and notice that this happens a couple of times as well. So here's carbon being stripped off this molecule, carbon being stripped off this molecule. So we go from a 6-carbon up here to a 5-carbon to a 4-carbon molecule. We also have reduced NADH a couple of times, 1, 2, and if you notice it over here, another one, overall theme to be kept in mind. And we've also created some ATP, 1 molecule of ATP. There's a little cycle here where GDP, guanosine diphosphate and guanosine triphosphate, just basically rotate around that phosphate to an ADP molecule, creating ATP. And FADH2 is created as well, another electron carrier. So this is a reducing agent that picked up some electrons, and it will be used later on. Ultimately, you have to keep in mind that for each molecule of acetyl coenzyme A, we create 3 NADHs, 2 carbon dioxides, an ATP molecule, and an FADH2 molecule. Notice that the 2 carbons that came in, they're denoted in red here. The red stays with the carbon chain, and the carbons that are pulled off are what I will call the old carbons that joined in back here, the blue ones. Notice the blue ones. And they are the ones that get yanked off of this chain. And then the red ones assimilate into the newly formed oxaloacetate, and they become the old ones for the next go-round. So again, it's a constant cycling of carbons throughout this whole thing. Now, again, keep in mind that's for every 1 molecule. So remember, there's 2 molecules of acetyl coenzyme A that come into this process. So that would mean a total of 6 NADHs, 2 FADH2s, and 2 ATPs being produced. Another checkpoint. I'll give you a few seconds here. I'll come back. You guys can hit pause and see if you can answer these questions. Okay, so continuing on here, we've got glucose, which has been changed into 2 molecules of pyruvate, went across the membrane, became 2 molecules of acetyl coenzyme A, giving off 2 molecules of CO2, and then acetyl coenzyme A entered the citric acid cycle, and we ended up getting rid of the rest of our carbons eventually. In the process, we've created 2 ATPs here and 2 ATPs here. Not really a huge yield out of this whole process. To give you a concept, we need 70 million ATP in order to power 1 heartbeat. So we're really not creating much energy here with 1 glucose molecule. However, what we have created a whole bunch of is these electron carriers, NADH and FADH2, here and back here in the process of glycolysis. Okay, so here's chemiosmosis. If we keep an eye on this here, this is a mitochondria down here. And if we look at it, there's all kinds of little folds and membranes in here. The inner part of this inner location is called the matrix. That's this word over here. The cristae are the little folds themselves, and there's an inner and an outer membrane. What's happening here is that this yellowish area down here is the matrix itself, and this is an inner membrane right here. And this inner membrane is between the outer membrane would be out here. So there's this area, this dead space in here called the cristae. So here's what happens within that membrane. There's those phospholipid bilayers that we should be able to recognize by now. And inside that mitochondria is NADH coming along, and it's carrying the electrons from the food. And what it does is it dumps its electron and hydrogen over to a protein complex, which is usually labeled 1, 2, 3, 4, and then there's some cytochromes involved here as well. These proteins are called the electron transport chain. And what they do is they rip electrons off of this NADH and the hydrogen, and that would mean that it is losing an electron, becoming oxidized. So this thing acts as this molecule here acts as a reducer at first. In other words, it gains an electron at first. This protein gains the electron from NADH, and then it passes that electron along, so becoming reduced at the end. So it becoming an oxidizer in the end. So it acts as a reducer and then an oxidizer. This acts as a reducer, accepting an electron, and then an oxidizer passing that electron along. Hydrogens are dumped across the membrane in this process. So they grab onto that hydrogen from NADH and eventually FADH2, and they dump hydrogens across the membrane. Ultimately, the idea here is that this electron keeps traveling down, or these series of electrons that keep dumped in here, keep traveling down the whole thing. Their ultimate end here is oxygen. And if you can remember, oxygen is your highly electronegative atom, and it is the one that kind of drives this whole entire process. It keeps the electronegativity so high that the electron has no choice but to be pulled along all of these protein chains. So oxygen joins in and it uses those electrons along with some available hydrogen, and it creates, with this last little step here, creates water. In that process too, we're dumping hydrogens across that membrane, and what we've done is we've created all kinds of hydrogens at the very top of this diagram at this point. Then what needs to happen is ATP synthase has to come along and pull those hydrogens back across the membrane. That's chemiosmosis, and I will show you a video on that in 1 second. Again, the diagram in the book is that this NADH drops off the 2 electrons, and those electrons go in from reducer to oxidizer, reducer to oxidizer. Each one of these act as a reducer and oxidizer. So all these protein complexes are labeled within here. They all have their own names. But eventually the thing that's at the very end of the whole chain is this oxygen waiting for the electrons and basically pulling the electron along the whole chain. And keep in mind that as each step goes on, hydrogens are brought across the membrane and across the membrane and across the membrane. And we end up creating a very, very high concentration of hydrogen. I'll go back 1 slide. The overall idea here is that you've got all these hydrogens. They cannot pile up on this side and create an imbalance across a membrane. If you remember osmosis, they're trying to keep a balance on either side. So what ends up happening is all these hydrogens are over here. They need to get back across the membrane. They're not capable of going across the phospholipid bilayer, and ATP synthase facilitates that passage. So we'll see how that works in 1 second. So here's that closer look at ATP. I have a link on here. I'm just going to go straight to the YouTube video itself and show you that. Oops, there it is. All right, so here's this concept of this ATP synthase molecule. And again, here's all these hydrogens up here. And the hydrogens are buried at the top of the membrane. This is our phospholipid bilayer design here. And this ATP synthase is a very large complex of proteins. It has different parts to it, which they'll label on here for you. And you can listen to the video later on. I just kind of wanted to walk my way through it here with you. These hydrogens come in, and what they do is they have a channel here to come in. They polar bond with parts of the proteins themselves, spinning this rotor head over here. And then as it spins around, it has a little exit gate, and that exit channel allows the hydrogens to come across the membrane. So overall, the hydrogens are being poured across the membrane, but they're spinning a rotor as they do that. It's a pretty fascinating process. Now, if we take a closer look at it from above, which we will here in a second, the rotor is spinning a shaft, which is then clicking these other enzymes into place down here. And if you take a look at what the ingoing and outgoing pieces are, here's ADP and P, which go into place, and then they get shifted, and when they get shifted, they're right in perfect alignment to form ATP, and then comes out. And it actually has 3 locations for that around the base of the molecule. So ATP synthase, massive molecule capable of churning out quite a bit of ATP as hydrogen goes pouring across that membrane. So once again, you can see the alpha helices and the beta sheets within here, and get the idea that we're made up of all kinds of different proteins in quaternary structure working together, very, very well orchestrated. They go in for a view here from the top, and they remove the rotor so that we can see the shaft a little bit better here. And we can see how the shaft has very specific alpha helices that act as kickers, like that one is a kicker right here, and it kicks the ATP molecule out of place. This one acts as a setter, so it puts ADP and P together, and each time it rotates through, it creates a bunch of ATP. I'll let you watch the end of that if you need to, but the idea here is that you need to have a whole lot of protons or hydrogens up here, and if it comes across, then it basically creates that spin. What's kind of interesting about this is it can work backwards too. If there's a lot of ATP out here and not much hydrogen over here, let's say that you store up a bunch of ATP and you don't need to use it, the rotor will work in reverse too, breaking ATP into ADP and P and dumping hydrogen back across the top of it. Pretty cool molecule, and I think that makes it a little bit more relevant. All right, continuing on here. So we've taken a look at that. Here's a checkpoint for you, and we're almost done. All right, so a little bit closer look at the overall thing here. Glycolysis, if you recall, required 2 ATP molecules to get this glucose to fall apart into 2 pyruvates, but it yielded 4 ATPs, so we ended up with a net of 2. This pyruvate, when it became oxidized across the membrane, creating 2 acetyl coenzyme A's, created 2 NADHs. We got our NADHs from back here. Kind of an interesting little side note here is that the NADH comes across that membrane, and those proteins that carry it across the membrane can either create NADH with it or FADH2, so we can have more hydrogens delivered as a result. These NADHs then are combined with 6 NADHs and 2 FADH2s from the citric acid cycle and getting 2 more ATP down here. And we end up with all of these NADH, FADH2s from all these processes dumping into the electron transport chain. This says about 26 or 28 ATP. It's not really known how many you're going to get out of each glucose molecule, because some of these NADHs that come across give you just 1 hydrogen. Some of them that come across give you 2 hydrogens, which can spin that ATP synthase a little bit more. And also ATP synthase, because it's a rotor, once you start it, it doesn't automatically stop on a dime when the hydrogen's out, so it continues to spin and kick out a little bit more. So again, this number is always going to be about 30 or 32. Overall checkpoint, I'll be back in a second. All right, the last thing that the book goes into, and I think it's worthy of note here, is that we have talked about ideally getting energy out of carbohydrates. And carbohydrates will go through this process, and they'll get glucose out of it, and they'll go through to pyruvate and acetyl coenzyme A and citric acid, and then they'll go through the whole thing here. And you'll get all that you need out of it. So ATPs and NADHs and FADH2s. If you go in and you consume proteins and you try to burn proteins for energy, notice that the entry point for this is simply down here. Those amino acids can be only reproduced or reorganized into pyruvate. And so we miss out on an opportunity to create some NADH and some ATP. Fats kind of have the same thing. You have your glycerol head here, which can be rearranged into this piece, and the fatty acid chains go into acetyl coenzyme A. The whole concept here is that your overall system or respiration is built to handle all 3 of them, but your best bet is carbohydrates. Entry points are at different places for proteins and fats, which don't produce enough NADH or FADH2. All right, so here's your overall homework. Be to review sections 9.1 through 9.4, and also read 9.5 carefully. 9.5 has to do with how overall this process happens if we don't have oxygen. So that's kind of an interesting key point there. I want you to keep in mind the relevance of this. I know it's really, really dry and boring stuff, but here's a picture of me and my nephew. This would be me, this would be my nephew. I don't know if you could recognize the difference there. I got him a shirt a couple years ago for his birthday. It said, "I'm with my BFF." And then I had this shirt on, just staged so that I could make a fool of him. It was quite fun. Going on internally, each one of us is this whole respiration process. And moving forward with this, we end up with our cells are made of different tissues and components, all the way down to the cellular level, and then each one of these cells contains mitochondria inside of them, and each mitochondria, and it depends on the cell, sometimes like muscle cells have thousands of mitochondria and fat cells have like 0. So it kind of depends on the cell and how many mitochondria you have available there. But each mitochondria is then doing a job, and inside that inner membrane space, you have that whole electron transport chain going on, pumping hydrogens across, bringing hydrogens back across with ATP synthase, creating your massive amount of ATP. So, wanted to give you that idea that this is happening on a very, very tiny, tiny scale, over and over, billions of times in each mitochondria inside of each cell, inside the trillions of cells that make up you. So, don't take it for granted. It's a pretty cool process, and it wouldn't work if all of it wasn't orchestrated absolutely perfectly.